AS Level Biology 9700, Paper 1, Variant 1, May June 2026. Second video on this paper, the question number 14 to 27. So first of all, I'm going to discuss the question number 14. And question number 14 is which graph represents the action of a non-competitive inhibitor. So you know that the non-competitive inhibitors are those which do not compete for the active site. So they can get attached on any part of the enzyme. So in this way, the shape of the enzyme is changed, due to which the rate of reaction is reduced. And in these four graphs on y-axis, they have given the rate of reaction, on x-axis the concentration of substrate. So in graph A, you can see that a non-competitive inhibitor when binds to uh, any part of the enzyme other than the active site, so it alters its shape. So the active site no longer functions effectively. So increasing substrate concentration cannot overcome this inhibition, because the inhibitor and substrate do not compete for the same site. As a result, the V maximum rate of reaction is reduced, so keeping the plateau of the inhibited curve lower than the uninhibited curve across all substrate concentrations. So according to this, you can see in the graph A, the rate of reaction has been decreased when there is attachment of a non-competitive inhibitor. Now we will move towards the B, why it is not correct. So you can see the rate drops to 0 as substrate concentration increases, which is incorrect, because this is not the behavior of the non-competitive inhibition. So B is not the correct option. Now C, you can see a linear relationship rather than hyperbolic enzyme kinetics. So C is not representing the correct answer. So now D shows the rate eventually reaching the same maximum speed as substrate concentration increases, which represents a competitive inhibition, not a non-competitive inhibition. So D is also not a correct option. Only A is there, which is the correct option. Now, question number 15 is glucose isomers in an enzyme are is an enzyme often used in commercial food production, and it catalyzes the conversion of glucose to fructose, which has a sweeter taste. The enzyme is immobilized on to a surface or within agarose beads as part of this process. The reaction is carried out between 55 to 60 degrees centigrade, which statements explain why carrying out the reaction at this temperature is beneficial. So the statement A is that immobilized enzymes contain more active sites at high temperatures. This statement 1 is incorrect. Why? Because the immobilization does not increase the number of active sites simply by increasing temperature. The activation energy of the reaction is more likely to be reached. So this is the correct statement. Higher temperatures apply more activation energy to the reactant molecules, increasing the proportion of substrate molecules with enough energy to reach the transition state. So this is the correct answer. Then the third, the increased kinetic energy results in more collisions between enzymes and substrate. This statement is also correct. Higher temperature increases the kinetic energy of both the immobilized enzymes and the substrate molecules, leading to more frequent successful collisions per unit time. Now, the fourth statement is maintaining this temperature is cheaper than carrying it out at lower temperatures. This statement is incorrect. Why? Because heating a system to maintain elevated temperatures that is between 55 degrees centigrade to 60 degrees centigrade, requires energy input and is generally more expensive than running a reaction at room temperatures. So this statement is not correct. According to this, we know that statement 2 and 3 are the correct statements. So we are going with the option C. So option C, according to this option C, there are two statements which are correct. So statement number 2 and statement number 3. Now moving towards the next question number 16. Now you can see the question number 16 on screen. Yeast cells have cell walls that contain glycoproteins. The statements describe the process that moves glycoproteins to the cell wall of yeast cells. So glycoproteins are packed into vesicles. Vesicles move to the cell surface membrane. Vesicles fuse with the cell surface membrane. Glycoproteins are released into the cell wall. So that is, you can see, they are packed into vesicles and then move to the cell surface membrane and fuse with, and then released into the cell wall. So according to this, you know that this is a process which is called as exocytosis. Exocytosis is bulk transport process where secretory vesicles fuse with the cell surface membrane to release their contents outside the cell. So C is the correct option. And you know that active transport is the process in which the molecules move from against the concentration gradient by using energy. So here is not only the movement, first of all, the whole process takes place, which is the exocytosis. Endocytosis is the movement in opposite direction. That is from outside the cell into the cell, and facilitated diffusion is again the type of active stance. So C is the correct option. Now moving towards the question number 17, and question number 17 is the diagram shows the structure of the cell surface membrane. So you can see which molecules regulate the fluidity of the membrane. So here they have given us the R is the glycoprotein, carbohydrate chain which is attached to a protein. So this is glycoprotein. And you can see the S is the phospholipid. Phospholipid bilayer. And T is cholesterol. Cholesterol which is located between phospholipid tails to regulate fluidity. T. And U is the intrinsic or transmembrane protein. This is the transmembrane protein. So we need the which regulates the fluidity of the membrane. So they are S and T. Phospholipid bilayer and this cholesterol molecule. So cholesterol strictly regulates membrane fluidity at high and lower temperature. The molecule responsible is T. So note on option, if this question, you can see that we have to say only about this, that is the, because T is only given in this option, and you can see the S is the phospholipid bilayer, and phospholipid bilayer and the cholesterol molecule both are responsible for this, specifically the cholesterol molecule, which is located between the phospholipid tails. So that is the reason phospholipids are also responsible for this. Now, question number 18. And question number 18 is, a student carried out an investigation to find the effect of surface area to volume ratio on the rate of diffusion of acid into an agar block. The student cut different sized agar blocks containing a pH indicator and placed each agar block into different test tubes. They recorded the time taken for each agar block to change color completely. So which row is correct for this investigation? So here you can see the size of the agar block, dependent variable is there, and then the variable that should be standardized during the investigation. So the size of the agar block, dependent variable, and the concentration of indicator in the agar block. So I would like to say that this dependent variable, according to this, the answer D is the correct option, because the time taken for the block to change color completely. So this is the dependent variable, and standardized during investigation, we have to keep the temperature standardized, or we have to control the temperature during this whole investigation, because we have to they recorded the time taken for each agar block to change color completely. So this is the time taken for the block to change color completely. This is B and D. Height of the test tube, this is not the you can say the variable which should be standardized, because it only shows the height, whereas we need to standardize the temperature during this. So D is the correct option. Now the remain behind the size of the agar block. No, we are not talking about the size of the agar block. We need to record the time taken for each agar block to change color completely. So A is not the correct option. B is also not the correct option. The size of the agar block is not the dependent variable in this investigation. Only D is the correct option. Now question number 19 is there. And question number 19 is, some plant cells are immersed in a range of solutions of different concentrations. In the more dilute solutions, the cell gained mass. Which statement is the correct explanation for this result? The plant cells gain weight in solutions of lower water potential. In this case, when these plant cells are kept into a solution with lower water potential, then what will happen? The water will move out. So water, this is not the correct option, because water will enter into the cells from solution of higher water potential. So they are talking about the lower water potential. So A is not correct. Second is, osmosis occurs across the permeable cell wall. B option is also incorrect, because osmosis occurs across the partially permeable membrane, not the permeable cell wall. Third is, the water potential inside the plant cells is higher than in the solution. So water potential inside the plant cells is lower than the dilute solution. So again, this is not correct. Now, the next is option D. The plant cells gain mass in solutions of higher water potential. So water, this is correct option, as when we will keep the plant cells into a higher water potential solution of higher water potential, then water will move from its higher water potential towards the lower water potential, and then the plant cells will gain mass. So D is the correct option. Now the question number 20 is, the table shows two stages of the mitotic cell cycle and structures that are found within typical plant cells. Which row shows structures that are present during cytokinesis and G1 in plant cells? So here you can see which row shows structures that are present during cytokinesis and G1 in plant cells. They are so the first option is centromeres, cytokinesis at G1, telomeres. So this is the correct option because centromeres are present during cytokinesis. So, and G1, during G1, the telomeres are there. So according to this, A is the correct option. And here we are talking about the plant cell. And you know the centrioles are not present in plant cells, so B is not the correct option. Then you can see the sister chromatids have separated before cytokinesis given. So according to this is not correct. So only A is the option which is correct. Now question number 21 is there, and question 21 is, scientists investigated the roles of two genes, gene P and gene Q, in stimulating mitosis in tumor cells. The scientists grew cells from different types of tumors and from non-tumor cells and measured the percentage of the cells that transcribed gene P only, the percentage of the cells that transcribed gene Q only, the percentage of the cells that transcribed both P and Q. And in bar graph, you can see on x-axis, the gene transcription has been on x-axis and percentage of cells transcribed genes is on y-axis. So you can see gene P has been gene P's represent that non-tumor cells which are with shaded area and tumor cells are without shaded area. So at gene P, you can see high expression transcription in tumor cells. Tumor cells is about 90%. You can see about 90%. And then non-tumor cells, they are only, again, that is, I think they are 90% and they are 15%. And in gene Q, you can see the non-tumor cells are at more than 90%, more than 90% or around 95%. And you can see this is, and the tumor cells are 20%. So gene Q high expression transcription in non-tumor cells is this, and so in P and Q, this is about only 10%, and this is about 15%. You can see that. So the options which can be concluded from the graph about the functions of gene P and gene Q. So you can see in this way the question. The option A is prevents mitosis, function of gene P prevents mitosis from starting. and a function of gene Q prevents mitosis from starting. So you can see the answer is not correct. A is not the correct answer. And again, prevent mitosis from starting. So again, it is not correct. Stimulates mitosis to start. Stimulate mitosis to start. Function of gene P. You can see the highest rate of the percentage of cells transcribing genes. So this is, it means it stimulates the mitosis to start. You know, during the mitosis, the cell start dividing. So this is correct for function of gene. And now the function of gene Q, you can see that non-tumor cells percentage is 95%. So this expression you can see the tumor cells here, high expression in normal cells and loss of expression in tumor cells indicates that it acts as a tumor suppressor gene preventing mitosis from starting. So it preventing the from so because in P, person highest expression transcription is highest. So tumor cells undergo uncontrolled cell division, and higher expression of gene P in tumor cells indicates that it acts as a oncogenic stimulating mitosis to start. So according to this, the C is the correct option. And stimulate mitosis, no doubt, for this is the function of gene P, but the function of gene Q is not correct over here. So C is the correct option. Now question number 22 is there. Which statement is correct? Adenine forms 3 hydrogen bonds with its base pair. Adenine forms 3 hydrogen bonds with its base pair. This is incorrect because adenine pairs with thymine or uracil only 2 hydrogen bonds. Adenine with thymine or only 2 hydrogen bonds. So this is not correct. The DNA nucleotide containing adenine is different from the RNA nucleotide containing adenine. DNA adenine nucleotide contains deoxyribose sugar, whereas RNA adenine nucleotide contains ribose sugar. So this statement is correct. Purines form base pairs with pyrimidines. This statement is also correct. Adenine and guanine, they are purines, and you know that. So adenine and guanine are purines, and pyrimidines are thymine and cytosine. So always purines pair with the pyrimidines. So according to this, statement number 2 and statement number 3 is correct. So D is the correct option of question number 22. Now question number 23 is there, and question number 23 is the diagram shows part of a polynucleotide phosphate molecule and pentose sugar has been shown in T. What is a possible base sequence shown in the diagram? So here you can see cytosine, adenine, and guanine. CAG. How? You can see the diagram shows a single polynucleotide strand with 3 nitrogenous bases attached to pentose sugar. The top base consists of single ring, which is pyrimidine, cytosine, thymine, or uracil. So the middle one, this one, and bottom bases consist of double rings. Both of they shows the double rings. So the consist double rings, which can be purines, adenine, or guanine. So adenine and guanine. So C is the can be cytosine, or they can be the guanine because they are double ring, or adenine. So CAG. So cytosine, adenine, and guanine. Cytosine, adenine, and guanine. So A is the correct option. All other options are not correct. In B, you can see that PAC means pyrimidine, purine, pyrimidine. This is not correct option. And you can see there is pyrimidine, purine, and pyrimidine again. This is not correct option. You know that adenine and guanine are purines, and cytosine and thymine are pyrimidines. So UAC, again, this is not correct option. So A is the correct option. Here you need to explain that the top base single ring shows the pyrimidines and double ring shows the purines. Now question number 24 is, which statement about the lagging strand in DNA replication is correct? DNA ligase catalyzes the formation of phosphodiester bond between short DNA fragment. This statement is correct because DNA ligase joins Okazaki fragments on the lagging strand by forming covalent phosphodiester bonds between adjacent sugar phosphate backbones. The statement B is DNA ligase catalyzes the formation of hydrogen bonds between nucleotides. Hydrogen bonds form spontaneously between complementary base pairs. DNA ligase does not form hydrogen bonds, it forms phosphodiester bonds. So this is not correct. C is also DNA polymerase adds nucleotides to the lagging strand in a 3 to 5 direction. So it is also incorrect. DNA polymerase adds nucleotides into 5 to 3 direction. So again, it is not correct. So DNA polymerase adds nucleotides to the lagging strand in a 5 to 3 direction spontaneously. So D is incorrect because synthesis on the lagging strand is discontinuous, not continuous. So according to this is not the correct option. Only A is there, which is the correct option. Now, question number 25 is there. The diagram shows a short part of the 3 nucleic acid strands. The diagram shows a short part of 3 nucleic strands you can see on P and Q. So here the P location of the transcription nucleus and transcribed strand they have shown. So you can see P is the location of the transcription is the nucleus. Yes, the option A is the correct option because you can see P is the transcribed and location of the transcription, you can see guanine, cytosine, adenine, uracil, thymine, adenine. So according to this, G pairs with C, A with U, and T with A, and A with U, and C with G, and C with G, and A with U. So A is the correct option. And whereas this second is the strand Q, you can see is C, T, T, A, G, C is identical to R, because this is the correct. So location of transcription occurs in the nucleus of eukaryotic cells where DNA is located. And R is, you can see the copy of R, Q. So A is the correct option. Now question number 26 is there, and question number 26 is the diagram show transfer sections of the part of the plant. Here you can see which most amino acids. So you know amino acids like sugars are transported through the phloem via translocation. And you know in leaf, the part 2 is the shows the phloem, and here is the 3, number 3 is phloem, and in root 5 is phloem. So which tissues transport more amino acids? So one is xylem, so it will not transport. Number 2 is phloem, yes, it will transport. Number 3 is also phloem, it will transport. And then number 4 is xylem, it will not transport. Number 5 is it will transport, whereas 6 is xylem, it will not transport. So according to this sequence is correct. So C is the correct option. So question number 27 is, the photomicrograph shows tissues in the stem of a grapevine. So you can see over here the transfer section tissue in the stem of the one of the cells in this transport tissue is labeled T. So you can see this is the T. Which statement about the cell labeled T is correct? The T has end walls that allow liquid to move by mass flow through the plant. Sieve tube elements, they are perforated end walls that allow organic fluids to pass through via mass flow. So it is the correct option. The T has lignified walls to provide mechanical strength to the plant. Lignified walls are characteristic of xylem vessels, not the phloem. T has plasmodesmata that actually transport liquids through the plant. Plasmodesmata connect sieve tube elements to companion cells, but liquid move through the main lumen and sieve plates by mass flow, not effectively through plasmodesmata. T transport water and mineral ions to the source. Phloem transport organic solutes like sucrose and amino acids from source to sink. not to the source. It means the statements B, C, and D are not correct. Only A is the correct option. Students, it was all about today's questions, which were 14 to 27. Now, please like, share, and subscribe my channel to watch more videos. Thanks for watching.